Thank you for inviting me. Today we'll, too loud maybe. Today we'll talk about the connectional architecture linking the human prefrontal cortex with the amygdala, and I will do so by using the connectional and anatomical knowledge we derive from the macaque-monkey model brain. This basically allows us to use the knowledge we have from track tracing performing macaque-monkey and try to check the reliability of our results first in the monkey using diffusion-weighted imaging and then in the human brain. Particularly, I will focus on the two main tracts which connect amygdala and prefrontal cortex, such as the ventral amygdala fugal pathways, the anterior limb of the ventral amygdala fugal pathway, and the uncinate fasciculus. Amygdala prefrontal cortex connectivity, especially the functional interplay of these two parts of the brain, has been implicated in a social cognition, in a cognitive task, as well as in the autonomic and emotional control, as well as has been reported to be disrupted in several psychiatric disorders. However, if we want to talk about functional interplay, we first need to have an anatomical understanding of the brain, to know the anatomy about the connectivity, so to have an idea about what is the anatomical connectivity before stepping into the functional connectivity. Right now, most of our knowledge is still based on animal models. As I said, track tracing studies have greatly contributed to our understanding of the connectivity between amygdala and prefrontal cortex, but the validity of these results is still unproven in the human. And most of all, track tracing studies, because laborious and because they require the sacrifice of the animals itself, well, they can't be performed in humans, but also when performing macaques, they don't allow whole brain analysis of the connections, of the connectivity, as well as they don't allow for longitudinal studies in the connectivity and its development. But by using diffusion-weighted MRI, we can create a whole brain mapping of the connection in the brain, and most of all, we can create comparative map between macaque and human brains and to check for similarities and differences across several species. So what we did, we used in vivo human brain from the Human Connectome project, and we used macaque post-mortem scanning. Using macaque post-mortem scanning allows us to have data quality and resolution comparable to the human in vivo imaging. And we have got our diffusion-weighted imaging data. And to deal for artifacts such as crossing fiber or kissing fiber or the artifacts which are proper of diffusion-weighted imaging, we use a Bayesian approach to inform our probabilistic tractography algorithms, and we reconstructed the connectivity of the two main tracts I mentioned before, so the connectivity of the ventral amygdala fugal pathways or the connectivity of the uncinate fasciculus, and we did this for both the human and the macaques using the same approach so that we could compare the results using the same technique. And then we use a connectivity profile matching approach to investigate differences between the two tracts and differences and similarities between the two species. The first thing which emerged from our results is that we can see here the ventral amygdala fugal pathways and the uncinate fasciculus. In red, yellow, the ventral amygdala fugal pathway, and in blue, the uncinate fasciculus. And in both the humans and in the macaques, they run parallel to one another for most part of their course. Throughout their course, they keep a specific organization, a specific medial artery organization in both species with the amygdala, with the amygdala fugal running close to the medial wall alongside subcortical structures such as the bed nucleus of the stria terminalis, the septum, the hypothalamus, and the medial preoptic area. And next to it, we have the uncinate fasciculus, which actually innervates more lateral regions. This is true, especially in the subcortical area, but as the two tracts enter the prefrontal cortex, the ventral prefrontal cortex, through the subgenual anterior cingulate cortex and the lateral orbitofrontal cortex, we can see has we can see how the two systems, the two connectional systems, merge together into a unified connectional system. So we have a separation at subcortical level, but then a integration and a convergence in the orbitofrontal cortex. Interestingly, by using the same technique, we could see that actually the organization of these two tracts connecting amygdala and prefrontal cortex is evolutionarily preserved across when we look at it into the two species, and the organization is very similar actually. We then wanted to move one step further and look at the connectional profiles, the connectivity profiles of both uncinate fasciculus and amygdala fugal. So we use a connectivity profiles matching approach, which allow us to plot our tract, the center of our plot, and then to look at to what extent each tract is connected to our region of interest in prefrontal cortex. And here you can see with the fan shape, the human connectivity, and overlaid on top of it, we can plot the macaque-monkey connectivity, and we can look for similarities and differences across the two species. We did this for the ventral amygdala fugal pathway, and we did the same for the uncinate fasciculus. We then looked at the similarities and differences across the two species to see whether we had artifactual projections or different projections in the human compared to the macaque. And we found actually that the amygdala prefrontal cortex connectivity is highly preserved, also in terms of connectional fingerprints between the two species. And then we checked actually a difference, because we would expect, if these are two different connectional systems, we would expect some differences between amygdala fugal and uncinate fasciculus. So we compared the two tracts, and we found actually some tract-specific connectivity patterns. If we look at each of them, we could speak a lot about this, but I will focus on the main one. If we look at the ventral amygdala fugal, both in its 3D reconstruction, we can, and in this connectional fingerprint, we can see that the amygdala fugal is located quite medially, and it has strong projections to area 25, to subgenual anterior cingulate cortex, which is weaker actually if we look at the uncinate fasciculus, which is approaching, as we say, the prefrontal cortex from a more lateral position. But if we then look at the uncinate fasciculus and its projections in the prefrontal cortex, we can see that it has strong projections to the ventrolateral prefrontal cortex, particularly to area 47, 47-12 in the macaque, so to the lateral orbitofrontal cortex, which are absent in the ventral amygdala fugal. This organization, this mediolateral organization, as shown also from our connectional fingerprint, could have interesting functional implication. Indeed, area 25, subgenual anterior cingulate cortex, has been involved in autonomic and arousal control, has been involved in motivational, encoding on motivational properties during decision-making or learning tasks, as well as disruptor in psychiatric disorders such as depression, for example. On the other side, the lateral orbitofrontal cortex has been repeatedly shown in both macaques and humans to encode for more sensory cognitive features in these tasks, for example, in encoding stimulus-reward associations in both species. So this tells us that actually, if we have two different tracts with two different routes are organized along a mediolateral gradient which connect amygdala and prefrontal cortex, and these tracts are, as we show, ventral amygdala fugal and uncinate fasciculus. Actually, we could have two tracts which are initially separate and carry different types of information, more motivational or more cognitive, to the prefrontal cortex or back to the amygdala. Since we can't address the directionality of the projection using diffusion-weighted imaging, we could have an initial separation of these two tracts, but then a convergence of the two tracts and an integration of the different type of information which is carried from each connectional system. We then wanted to expand our results. And please stop me, I don't have the time to go through all the system here, all these three systems, which these three tracks which project prefrontally. Stop me, we can talk about it. We basically use the same approach to reconstruct the anterior projection, the prefrontal projection of the internal capsule, the extreme capsule, and the cingulum bundle. And I will quickly mention that actually we could find similarities in the connectivity profile between macaque and humans in also these three systems. So to conclude, I hope I convinced you that by using diffusion-weighted imaging and probabilistic tractography, we can reconstruct connectivity in the brain, and especially we can reconstruct amygdala-prefrontal cortex connectivity across several species. This allows us to use track tracing literature to inform and to check the reliability of our diffusion-weighted imaging results in the monkeys, and then use the same approach in the macaque and create comparative maps across the two species. I also hope I convinced you that actually we have these two tracks as reconstructed with diffusion-weighted imaging run in parallel, and they consistently show this mediolateral organization, and that they merge together in the posterior orbitofrontal cortex, such as area 13, or rostral subgenual anterior singular cortex, area 25. They merge together into a single system, with the amygdala fugal being a more limbic, more medial tract, running alongside more subcortical region, and the unseen fascicle is actually a more laterally projecting tract carrying more sensory cognitive information to the prefrontal cortex. And this separation of the ventral prefrontal cortex into this subdivision, actually, of the ventral prefrontal cortex is not new. It was suggested already by Price and Hongur in early 2000, but also they started showing it in the late 90s works using track tracing studies. And it's not new, but what is very interesting here is that using diffusion-weighted imaging, we could actually reconstruct such subdivision in the prefrontal cortex, and most of all compare it between two different species to try to bridge the gap that we have right now in the anatomical understanding of the brain. To conclude, I would like to thank the people I've been working on, especially Lennart Verhagen, Rogier Mars, Jerome Saleh, Matthew Rashford. I would like to thank the Human Connectome Project Consortium, and I think to conclude, recognition should go to the animals and the contributions they are giving in our understanding actually of the brain. Thank you.
Yes, two comments. I wouldn't call it the amygdala-fugal pathway, because there is a connection between the amygdala. It's called the amygdala-fugal peduncle, but it goes via the just below the anterior commissures, and it goes into septal areas, nothing to do with the prefrontal cortex. Second, I wouldn't call it amygdala-fugal, because there are connections from the orbitofrontal cortex to the amygdala, so it's bidirectional. We know that. So, you know, maybe it's a matter of semantics. I would choose another name for that. So the second comment, I wouldn't use the human connectome dataset for this study. We try to do what you've done before on the human connectome dataset. The imaging from the connectome dataset is very poor for the uncinate, for any connections that goes to the orbitofrontal cortex, and that's because the signal of the diffusion data from the human connectome dataset on the orbitofrontal cortex is absolutely too low, is too poor. So the orbitofrontal cortex is not a major hub in the human connectome dataset. I wouldn't use it. We had to change completely the dataset. We couldn't see what we have seen on the 1.5 Tesla, the connection to the uncinate 20 years ago. So, you know, I strongly recommend that in fact, your images, when you look at the connections into the temporal lobe of the uncinate, they're very thin. I'm sure you're familiar with this, and you looked into that.
Yes, thank you for the comment. The reason we use the Human Connectome project data is because they were available by the time we wanted to start this investigation, and we are now going to collect some more accurate diffusion weighted imaging data and try to replicate the same results. This is also because we were aware of some of the limitation, especially in the ventral prefrontal cortex acquisition. This is why we try to reconstruct actually the connectivity profile of extreme capsule, which is more dorsal, and the cingulum bundle and the internal capsule as well.
You have a better data, so the fact that it's from the human cognitive data set doesn't necessarily mean that is the best data set for every track.
I perfectly agree on this.
My point is that for the ancillary fasciculus in Oxford, you acquire much better data set.
Thank you.
We're about 10 minutes over, so we have to move to the next talk. Sorry.